It's always good to see other friends from other parts of the campus at our conference. And actually, this isn't our usual time, but when we have a distinguished guest, we accommodate the time. And just for those of you who are not regular attendees of DMICE conferences, we're an informatics group, so we stream these, and we actually let people who are out there in cyberspace watching send us questions by Twitter. So we'll monitor the Twitter feed if any questions come in. I attended one of our conferences from Riyadh, Saudi Arabia last month and managed to tweet in a question. And when you do ask questions, if you can press and hold down the little button on the microphone that enables the audio for people to hear the audio, otherwise they can't hear it. And we also record these, and they appear on our website. So it gives me great pleasure to introduce Katerina Kechris, who is visiting from the University of Colorado, Denver, where she's an associate professor in the Department of Biostatistics and Informatics, and works with a colleague of ours, Larry Hunter, who I've known many years. She received her PhD in statistics from UC Berkeley, and then did a postdoc at the University of California, San Francisco. And she's going to talk to us today on regulation of the transcriptome.
Audio visual, does this work fine? Okay, great. Great. Well, thank you for the introduction, and I also want to thank Shannon for setting up this visit. It's been a pleasure to meet with some of you earlier today, and I look forward to talking to some of the others tomorrow. So today I'll talk about computational methods developed in my group to study the regulation of the transcriptome. But before I get into some of the details of the methods, I wanted to first introduce myself, for some of those that I haven't had a chance to meet. I have training in statistics, as Bob mentioned, and I'm interested in the development of statistical and bioinformatics methods. So it's my emphasis on high-throughput data analysis. And related to that, with my recent collaborations, their investigators are using multiple technologies to profile their systems, I've been involved in integration of different types of omics data, which poses challenges from the statistical and computational side. I'm also interested in the development of software methods so that some of these methods can be general for others. I typically work in statistical software R, but also in other programming languages. So that's my interest from the statistical and computational side. From the biological side, I'm interested in the processes that regulate transcription. So the different mechanisms, whether they're the role of transition factors or epigenetic effects or post-transcriptionally by microRNAs and other non-coding RNAs. And as you know, there are many different types of technologies, array and sequence-based, for obtaining genome-wide profiles of different maybe stages of these mechanisms, getting the expression measurements for genes and non-coding RNA, looking at these DNA sequence to look at binding sites for transition factors or microRNAs, the DNA methylation profiles, and other types of genome-wide data. So that's my interest, again, on the biological side, is these mechanisms that are important for transcription regulation. So you've got the statistics, you've got the biology. What brings it together are the collaborations I have with biomedical investigators. I'm currently at a medical campus similar to this environment, so very close work with biomedical investigators. I've highlighted two of my main collaborations throughout the years since I've started there. One is using animal models and behavioral models for alcohol use disorders. There's several investigators in Denver, in Boulder, Boris Tabikoff, Paula Halfman, Richard Radcliffe, and Tom Johnson. And I'll talk a little about that later today. What I want to talk about is another main collaboration with lung disease, chronic obstructive pulmonary disease. This is large human genetic epidemiology studies where I'm involved with different omics profiling, proteomics, metabolomics, and trying to integrate those different molecular profiles. So as you see, these are very different disorders, but there's a common theme. One is sort of the bad things we do to our body, whether that's excessive drinking or smoking, sort of the environmental exposures. But the common theme on my end, I think, is the complexity. These are both complex diseases at the molecular and genetic. These are polygenic disorders, multiple pathways involved. but also complex diseases to characterize, whether it's acute or chronic effects of ethanol exposure, or for COPD, there's different types of clinical metrics, spirometry-based, or CT scans. So there's also complexity to define the disease, and there's work involved in better characterization of the complexity of the phenotype. So those are common themes for these collaborations. I just wanted to mention a few others, wide-ranging from the role of HPV in development of cancer to fertility to obesity and diabetes. Again, I play a similar role where I'm involved with the regulation of the analysis of the transcription regulatory data. I wanted, since there's such a strong alcohol research group, I wanted to highlight some work with alcohol researchers in Colorado before I get into some of the methods that I'll talk about. And for example, this is a study that's now been under revision that's looking at selected lines for alcohol preference and looking at expression profiles during the selection stages. So we can look at the expression profiles in conjunction with the alcohol preference measurements. But throughout the years, I've worked on other alcohol consumption intolerance models and analyses of the gene expression data, again, contributing to whether it's microRNA sequencing analysis or transcription factor binding site analysis. Also some work with Tom Johnson in fetal alcohol syndrome models, again, using mouse strains. And sometimes this has sparked methods development to look at, in this case, it was a multi-treatment, so multiple strains, multiple brain regions, and looking at expression at different times after exposure. So to develop general models for that purpose. And I've also contributed to the Phenogen database website, which is to study complex diseases, complex traits, and contributed to the modules in the database for related to expression analysis and transcription regulation. So that's a snapshot of my interest and what I've been doing for the last 8 years. And I find the collaborations is the most enjoyable aspect of my work. Just pause a second. We'll now switch gears if you have questions to some of the methods developed. So I'll go into more detail about some of the methods developed in my group related to the regulation of the transcriptome. And I'm using a very simple central dogma diagram, very simplistic, to show the information flow from DNA to RNA to protein, and then how these methods play into those different processes. The first method is related to the role of transition factors, so these important regulatory proteins to initiate or repress transcription, and looking at binding, because the recognition of transition factors of their binding sites is important for their regulation of regulating downstream targets. So I'll talk about analysis of in vitro binding data. Then that will lead to an analysis of in vivo binding data. So now there are technologies to examine where transition factors are binding across in the genome. Okay? But this is genome-wide data, and it can also be used for the analysis of DNA methylation based on arrays. As I mentioned before, a lot of the labs I work with are using different technologies to profile their systems. So I've worked on development of methods that integrate different omics level data at different stages, whether it's DNA sequence, binding data, or the actual expression measurements. Talk a little about that. And finally, I'll look at post-transcriptional processing. So after the RNA has been transcribed, there are other types of regulators, particularly non-coding RNA like microRNA. So I'll spend some time about this is my most recent work, and I'll come back to an alcohol example. So I'll go through the different methods in this order, and they also increase in terms of complexity. In terms of in each of these, there's a motivating example based on a collaboration, and it starts from yeast and bacterial in vitro binding examples to using Drosophila as a model system for studying development, and finally to the behavioral models in alcohol, just to look at microRNAs. So I'll start with the first one, which is called, the method is called SULDEX. And just to review, transcription factors are important proteins that recognize they are DNA binding, so they recognize their binding sites near target genes, usually in promoters. And here's an example of some binding sites for a transcription factor in yeast called GAL4, responsive to lactose-rich conditions. And following this recognition, they interact with the RNA polymerase and different cofactors and multitude of proteins to help initiate or repress the transcription of a target gene. Okay, so that's a role of transition factors. As you go to higher organisms, so in yeast they're very well studied. A couple hundred are very well studied. You go to higher organisms, they're maybe estimated up to 15 to 1,700 different DNA binding transition factors. And there are efforts to try to elucidate their binding in the genome. But understanding their particular binding preference, like you would see here, there's still a lot more work in that area. And understanding their binding specificity helps to understand where they bind in the genome. So it's a problem of important because you can look at targets of particular transition factors and pathways once you have this information. So we used high-throughput sequencing approaches to perform a quantitative analysis and then developed sort of a model for estimation of the transition factor binding energies using some biophysical properties. So before I talk more about our method, I wanted to mention that there are sort of smaller scale approaches to study transition factor binding in vitro, gel shift assays where you can look at predefined sequences and find their binding potential. There's also screening-based approaches like SULDEX, where you screen a random pool of oligos and try to, in the screening assay, identifies those that have higher binding. So these trying to find the highest binders. There are also moderately binding types of sequences that may not be captured in this approach. So in the last few years, SULDEX type approaches have been scaled up. So now we have next generation sequencing, so we can now perform parallelizable, looking at multiple transition factors, and looking instead of a predefined small set of a few dozen, looking at thousands and hundreds of thousands of possible oligos that may be binding to a transition factor. And they have names like bind and seq or HT SULDEX, SULDEX seq, so it kind of gives you an indication that they're building up on the SULDEX approach. So we've worked in this area as well, and this is the method is called SULDEX, Simultaneous Ultra-High Throughput Ligand Dissociation Experiment. It's a mouthful. SULDEX is a nice abbreviation. And the idea is that you can mix random oligos first without a transition factor, get a baseline for what the frequency of those oligos are, and then you include the transition factor and extract the bound oligos, and then sequence those. Okay? And then you can compare relative frequencies, and then you can identify the type of binding sites that are preferred by the transition factor. We used some biophysical principles to model the frequencies to estimate these binding energies, KDs, using a Bayesian model and Markov chain Monte Carlo to obtain posterior estimates of the mean binding values for each oligo. Here's just an illustration of the data, sort of hypothetical cartoon here where pre-binding, this is before the transition factor is included. Here's just a list of some of the oligos. Obviously, this list continues. You have counts before, and then once the transition factor is included. And most of the time, you see this variation within the noise between the two, between pre and post, or even high levels that are similar. Sometimes you see reduction. I'll talk about that later, why that may occur. But the interesting cases are like this, where you see an increase. So once the transition factor is added, that particular oligo is enriched, indicating that it's possibly binding or has a stronger preference for that binding sequence. We can use some standard count-based models, multinomial models, where you have a parameter for the frequency of each oligo. So we can have the pre-bound frequencies and the post-bound frequencies and the overall count. That's the basic assumption we use for the counts. And it's a common distribution for counts. But we wanted to incorporate the biophysical properties. Okay, so something we know about how transition factors are interacting with their DNA recognition site. So here's an equilibrium reaction with a very, again, we're taking them just by themselves. We had a transition factor on a site, a sequence site. So here's free transition factor in site, and here's the equilibrium reaction where you have bound sites. And then you go back and forth with a certain rate of binding and dissociation. Again, since I'm a statistician, I like to express this in probabilities. You can express this as a probability of binding, just the concentration of the bound versus the total. Okay. And there's this equilibrium constant, which is the quantity of interest. That's the rate between the dissociation and the binding. And that's this ratio. So putting this all together, I'm skipping the math, just hope you trust me, this can be expressed as the same probability as a function of two quantities, the transition factor concentration and this KD. And so that's the parameter of interest for each oligo that tells you what strong binder it is. Okay. And for those of you with some physics background, the KD can also be expressed as a free energy of binding, a delta G term, using the simple equation. So we use these interchangeably. You can go back and forth using this set equation. So putting this all together, here's an illustration of our model. And I know there's a lot of equations, and I heard you lose half your audience for every equation. So hopefully that's not the case here. But I wanted to highlight, I'll highlight the important parts of this. It's just a graphical model version, if it's a computer science world, of the model. So we have our data. These are these pre-counts, the counts before and after. I mentioned this is multinomial. That's the data we observed. That's shaded. We also have some reference KDs for at least one oligo. That's important for calibration. If not, you get relative metrics. So this is important for calibration. Otherwise, you'll get a relative delta G. So that's the data that's observed. And then there's parameters of interest that I've talked about. There's the transition factor concentration. There's these pre-bound frequencies. We need to know sort of what's baseline. And then either the delta G or the KD. You can go back and forth. And all the other intermediates are just set by deterministic equations. So there's no probabilistic relationships there. So we just need to go through these intermediates. So use the data to estimate posterior probabilities of these quantities. And these will tell you about, again, what kind of binding site does that transition factor prefer. And again, that helps you understand possibly its targets in the genome. Now, there are some limitations to this approach. If there are low counts, so sequencing data, if you don't do enough sequencing, you may have low counts. Remember, we're looking at thousands of possible oligos. So there could be summations, could be inaccurate due to low frequency counts. And if you have high transition factor concentration or low transition factor concentration, that could be a problem. And you can think of it, if you have a lot of transition factor concentration, there's a lot of them floating around, and they're going to bind kind of not specifically. Okay? If there's not enough, then they will bind to only the highest binders. And if you're familiar with position weight matrix models, those assume there's a low transition factor concentration. So it's really biased towards the highest binders. And we wanted to be a little more flexible. We wanted this to be a parameter in the model so that we could look at more intermediate types of binders. So we have a model extension that pools information across all the data. So now you have a contribution based on every position and every nucleotide. So you have some kind of optimal binding energy, and depending on each nucleotide at each position, you have a contribution to the energy. And you can look at higher order terms. We're not doing that just to keep things simple, but you can look at pairwise. It is known there are known to be interactions between positions in a transition factor binding site. So you can expand this as well. So that's the model. I'll discuss some results. There's more in the paper. Where we had here two transition factors, again, in these I mentioned yeast and bacterial. So relatively more studied in bacteria and yeast. So we had some known KDs, and we could simulate a repertoire of sequences that you would observe in one of these types of sequencing studies. The pre-bound, let's assume equal frequency, and post-bound, you can use these known KDs. And start to vary these, first of all, how much sequencing you're going to do. So this is an experimental design issue. What kind of precision do you have depending on how much sequencing you do? And also varying the effect of the transition factor concentration. And I may have forgotten to mention that you can't, in the experiment, you can't necessarily set that precisely. And that's why it's important for that to be free. You can do some follow-up assays to try to deduce what the concentration is, but it's not something you can set precisely. And that's why we think it's important to have it as a free parameter. And we can just look at the root mean square error of our prediction. So how well do we predict the known KDs? So here's an example of one of the transition factors. The x-axis is the average count, so this is how much sequencing. So the more sequencing as you go to the right. The y-axis is this root mean square error, so that's how we perform. The smaller, the better. That means we're predicting better. And the two models, this basic model and then this model, the generative energy model that pools information across positions, is in red, so blue and red. And so one trend you see is definitely more sequencing, more data, the better. It's most always the case. So that was just confirmed here. And as you increase the transition factor concentration, you also increase the error rate of that basic binding model, which is something we hypothesized that would happen because this basic model is more sensitive to concentration, where the other one, the y-axis is changing. So you can see the red bars are more stable. They're less sensitive to that. Particularly, it's something you can't always control. And in this case, this extension model that pulls information across the positions and sites also performs better. But this is not always the case. I can't, for the sake of time, I can't show all the results. But for other transition factors that bind in a different fashion, you actually get sometimes the basic binding model does better. Okay. Now, how does this relate to the actual binding site? Again, the purpose of this is to understand the specificity of a transition factor, and then you can use that information to look at possible binding sites in the genome, okay, and look at possible targets. So here are the posterior distributions of these position-specific energy terms for Lu3. Here's a cartoon of its structure with binding to the DNA. And here's a consensus binding site. So here we know the truth. They wanted to see how well we do. So the columns are different, ACGT, different nucleotides. The rows are the positions, 1 through 10 across. And the model actually tries to find the optimal nucleotides that may be occurring. So sometimes some bases, A is not really meaningful if you convert it to an A, that it's just not selected in the model process. So the optimal binding energies here, C, which you would see in the consensus. And then if you have a nucleotide substitution, G or T, you can see the effect of that. So T is very deleterious. So you have a high additive component. So if you observe a T there, it will affect the interaction between the transition factor and the DNA. So this is probably the largest one. In other cases, here position 3, you can interchangeably go between G and T and not have that much effect. So that's how you can summarize and interpret the outcomes of this model. And you can move along that and see the effects. So that relates it to the actual binding site. So just to move along and review, this is a method I forgot to mention. This is with David Pollack's group in biochemistry, molecular genetics in Denver. And this is a software in C that's available on his website. And we've come across some issues with some of the existing HT-Celix or Celix-seq type data, is that the pre-bound frequency, so up to now I was just saying, oh, well, we'll assume they're equal frequency, but there's biases in the sequencing, where could be some secondary structure that affects which oligos are sequenced more than others. So we found orders of magnitude differences in some of the oligos in the pre-bound frequencies. So forget the transition factor's not even there. You expect uniform distribution, but there's very large biases. So we tried to account for that, building a third-order Markov model, so to try to get more accurate frequencies. And you can see there's, based on some of these structural issues, that you get some GC bias, so they're not evenly sequencing the pre-bound oligos. So that needs to be accounted for, or else your later estimates are biased. And this approach can be used for experimental design issues, dependence, the count. You could see as more data, how much sequencing would you need to do, and what kind of concentration would you need. So, and what's the length of the transition factor binding site. So it provides a platform for exploring all of this. Okay. So that concludes the introduction to the SULDEX method that, again, I mentioned is in vitro binding. And we had a yeast and a E. coli example. So now I'm going to increase in complexity. So I'm going to start looking now at in vivo binding of transition factors genome-wide, and there's a fly developmental model for that motivated that problem. So the method is called COMP. And the motivating example is with a collaboration with the Kornberg lab at UCSF. They look at fly development. And the pathway they're looking at is Hedgehog. Here's Hedgehog. And there's a downstream transition factor, Ci, that's involved in activating several pathways involved in development, particularly wing development. And the Hedgehog pathway is also important, not just for development. There's a homologous pathway in human, sonic hedgehog, and that's also been implicated in the development of cancer. So there's some disease implications too. So Ci is right here. That's the transition factor of interest. And you can see when you have a wild-type wing compared to a mutant Ci wing, it's smaller, there's some veins missing, and all that affects the function of the wing. So CI is an important component of this pathway and proper regulation of the downstream targets. So they were interested in identifying targets of CI, and they had performed a in vivo binding assay, so based on DAM ID and tiling, and I'll go into that in the next slide. And my role was predicting regions of high CI binding levels in the genome. Problem is that there were very strong spatial dependencies as you moved along the genome with this type of data. And it was important to account for that, otherwise we would get a lot of false positives. So this is a review. Maybe you're familiar with chip methods, array, chip, or chip-seq. So these are high throughput methods for the isolation and identification of DNA sequences bound by a DNA binding protein, for example, transcription factor in this case, across the genome, and this is an illustration of the method. The chip method here is based on an antibody approach. They had an alternative called DAM ID. I can't get into the details, but in that case, antibody is not needed, and that's the advantage of using that. So that's an alternative here. And now, instead of arrays, often sequencing is used. Okay? But the end goal is similar in that you're trying to find peaks along the genome that are possibly targets of this transcription factor. So for the array data, this is what they had generated. Here's this illustration of the data. You have the chromosome, you have probes across, and there are chipped DNA-bound regions, or DAM ID, that are obtained by those are regions that are bound by the transcription factor. And then there's some controls that should be randomly distributed in the genome in blue. So the peak analysis would be finding series of enrichment of bound versus control. So you can look at a ratio, and here are much more frequencies of the signal for the chip enriched versus the control. So in the analysis method, I mentioned the challenges of the spatial dependencies. The bound, this DNA sequence that's obtained is much longer than the probes themselves. So you can get spatial dependencies there. And the methods that have been developed, for example, they may rely on hidden Markov models that try to capture some of that local relationships, but they can't capture too long range. They tend to be 1 or 2 or 3 positions. And moving averages are simple, but more intuitive. And I'll just show you an example. Here's, let's say, the original values of probes, some ratios, and you're just moving along, calculating the moving average as you go along and find high values. So much more simpler, but also intuitive for interpretation. Now, the limitations of the moving average is they are not very convenient for alternative types of analyses. And this came out because I had another collaboration where it was a large human cohort. You had lots of clinical variables. You need to adjust for those. So having more complicated modeling was important, or maybe non-parametric statistics or more complex studies. The moving average was based basically on this ratio. The other issue was the correlation among the probes. And this is not corrected. It could inflate significance. So I'll show you here an example from the fly data. Here's the lag of the probes. So you have 1 lag, 2 lag, and you can calculate the correlation. So of course, at the probe itself, at 0 lag, it's 1, the y-axis, and the next lag 1, you have correlation of 0.6. So it's pretty strong, and it drops off eventually. For DAM ID data, this is particularly stronger because the DAM ID method the regions are longer than chip. Okay, so it was an issue for their data set that I needed to address, okay, because if that was not addressed, we would get lots of predictions, and they tended to be false positives. So we needed to make these adjustments. So what I worked on was a generalized moving average, taking the ideas of the moving average, since they're intuitive and help, you can explain them much easier than a hidden Markov model. So the idea was to use a p-value at each probe for a particular test, and then you can incorporate the complex analysis, whatever it is, whether it's a human study, you need to make adjustments, or a non-parametrics test. You can also account for direction using a one-sided test. Now, it's not appropriate to take averages of p-values since they're distributed between 0 and 1. So you can draw from the meta-analysis literature. So here I have one example. I tried different types, but they're called combined p-values. So the idea is you take a p-value at each probe, and you can convert this using the Gaussian cumulative distribution function, and then that can be averaged. Okay, so this is called the Stufer-Luptak test, and there's some alternatives that I also tried. So now I have a moving average of something I can actually calculate a moving average of. So I converted the p-values into something that you can actually take an average of, and then use standard theory for averages. Now, the other problem was the correlations. And it depends on the type of test. So I'll just show you this particular one. For the one that I just described, where you converted the p-values into q or z values, you can think of them as z values. Well, we have this correlation matrix. They're highly correlated. And again, if you don't correct for this, it causes problems. So I won't go into the math, but after some transformations and some matrix algebra, you can make them so that they're now independent and follow standard normal distribution, and then use all the standard theory. Okay, so again, another transformation to make them usable. So that was the crux of the approach. I also tried alternative combined p-value statistics and alternative corrections, but this shows you the general approach. Once you have these combined p-value averages, you can, as you move along the genome, you can look at the significance, you can look at this moving average and define peaks. Also correcting for multiple testing, and for now I just use a set cutoff that the probe should be within a certain distance, although I've relaxed this in future work. So now you can define a region of the genome that shows this trend associated with your test of interest. So I have here a diagram showing that. You have these adjusted p-values moving along, and this would be the window that shows the biggest trend. Okay? So whatever the test you are performing, this is all in the same direction and shows a string of values that are consistently significant. Now, to evaluate methods, an important problem of bioinformatics research is we have ideas, but you can always don't know if they're working because you don't always have gold standards. Unless, I mean, you can do simulations, which are a great start. So trying to find how to evaluate this approach was important. I worked closely with the lab there, and they had a list of predicted gene targets of CI. Some of these very well curated from the experimental literature, and others were from expression data, maybe predictions on their part. So we can examine the predicted peaks, whether they're close to target genes, and look whether that was expected in the total genome, because of course these peaks, by random chance, they may be occurring near target genes. So you need to correct for that. The other evaluation was, fortunately for CI, there is a consensus binding site that's GT rich, and we could examine the currents in peaks, of course scaled by the length, because this is a pretty short motif. It's going to occur randomly in the genome, so you need to correct for that. And here's some representative results. There's a lot of lines here, but I'll break it down. The x-axis is different significance thresholds. So as you move to the right, more significance correcting for multiple testing. Y-axis is these two metrics, target gene enrichment and motif enrichment. And the lines are the different methods. So I'll highlight first the bottom lines. Okay? This is when no dependence was built into the method, okay? Using both the methods that I propose and some of the existing methods in the literature, a basic moving average and an HMM. And these both, when you don't account for dependence in the spatially across the genome, you get less target gene enrichment and less motif enrichment. And what's happening is, even at this very stringent threshold, you're predicting thousands of peaks, orders of magnitude more than if you had the dependence. So picking up a lot of noise. And that's reflected here is that there's picking up a lot of random peaks in the genome that are not near at least the known target genes. So that's why these metrics are reduced. Once you include the enrichment, you get much more, wrong direction. Much more enrichment. You're getting peaks near genes that are more meaningful based on what they've provided, more motifs. So that it's, and you're just making less predictions. So just fine-tuning the predictions. Among the methods, the ones that I proposed are in the circle and the triangle, and those perform better for the more significant thresholds. This is a basic moving average. And later, for least stringent thresholds, they're more similar with the slight advantage of the basic moving average. But like I mentioned, the basic moving average doesn't allow for more complicated models. It's just ratios or maybe a t-test analysis. So this is just an illustration, and we tried it on other data sets and different examples. Now, this method has evolved. Again, I was interacting with a group of lung researchers on campus who were looking at a lot of epigenetic effects, so environmental effects to explain, for example, asthma. So they had an asthma study. And they were doing epigenetic profiling using methylation chips. This is just a picture from the Illumina chip that they use, where you have probes for the methylated state and the unmethylated state. And they like this idea, but we needed to work on making it more adaptable. For example, probes are variably spaced with methylation data. These chips because they're at CPG loci that are not the same location or the same spacing. So you had to account for that. Being more dynamic and flexible to define your peaks, and assigning a p-value for the actual peak itself. That was a limitation before. So, and doing a multiple comparison at the peak level, not the probe level. So those are the ways we adapted it to make it more suitable for the type of methylation chips. And also these were human studies. This was the kind of study that I mentioned you needed to adjust for clinical demographic covariates. And this is a Python script that's at the GitHub site that's available. And I've seen it used in, I mentioned I'm involved in COPD research in some COPD applications as well. So if there aren't any questions or Twitter questions, I can take a pause if, okay, just moving along. So I talked about nitrogen factors and methylation, and again, those are important mechanisms that can regulate transcription. The third method I'll talk about is data integration. How can different data be combined for one study to try to get better inference? Okay? So the example here, I have data integration both at the DNA level, the transcription factor binding level, and also the expression data. And this approach is called LC-Mix. And it was used the Hedgehog Ci study as a motivating study again, although it's had other applications. And I just wanted to highlight, this was a PhD student working with me who is on an LM training grant. I just wanted to highlight similar, I think, to the one that you have here. And he just graduated last fall, so I wanted to acknowledge his work on this. And the goal was the same. How do I identify CI targets for this pathway? But now we have not only the binding data, there's expression data, there's several mutants of the CI, this pathway actually, some earlier proteins involved in the pathway. And also sequence conservation. So in this case, this is development. It's highly conserved among fly species, and there's a fly species alignment with mosquito and honeybee in UC Santa Cruz. So you could look at conservation as well. And the idea was to develop an integrated model to use all that information to obtain targets for CI. So the approach we used here was a hierarchical mixture model. And the hierarchical part is that this is you have the observed data here, binding, expression, conservation. And what's unknown is whether a particular gene is a target or not. But whether it's a target or not, then you may expect binding or not binding, or expression changes or non-expression changes, or differences in conservation. So that's the hierarchical part. And you can look at different organizations, different topologies. This one treats the different processes in the same equally, where this approach tries to use some maybe the biological knowledge that if it's a target of CI, the hedgehog pathways, then it would maybe have CI binding, and if the target gene, and then that expression of the target gene changes, and then that expression may be selected on, and it may be reflected in the conservation. So that's more looking at the process and the flow of the process. So you can have different topologies, and these are the two main topologies we've looked at. These are part of a Bayesian belief network, so you can think of more other kinds of topologies, but we focused on these. The mixture part is that you expect different distributions. So here are known targets that were collected from our collaborator. Binding is shifted for the known targets, and the binding is for non-known targets is centered around 0. Okay, so this is called a mixture model where you have two distributions. So each of these you can have different distributions, whether you're bound or unbound, differential expressed, upregulated or flat or downregulated. So you can have three mixtures. Okay, so there's flexibility in allowing for different types of different numbers of distributions to reflect the different states. So model filling was performed using expectation maximization to fit each of the individual data types, and then looking at an extended EM algorithm to pick between the topologies. We don't know in advance what may be better fit. And there's model selection procedures for that I won't go into. So first, use some simulations but to try to mimic the CI binding data. And these are just a summary of those main results, where if the predictors were all equally good, the layered model, which treated them equally, tended to do better, which is expected. If you had a poor predictor, for example, conservation is not a particularly good predictor for CI. It has some information, but other types of genes are conserved in other developmental pathways or other kinds of pathways. CI in the Hedgehog pathway is not the only reason you would see conservation. So it's not saying conservation would not necessarily be the best predictor, but it is informative. Okay? So a chain model where maybe the poor predictors are at the end would perform better. And then we looked at some of the model fitting approaches. This is more the bioinformatics, is how to implement this in the best way. I won't go into that. So here's an example of one of the data sets, the CI1, which was a motivating one. So the x-axis is just the percent predicted. So we have about 10,000 genes. And depending on different posterior probability cutoffs, you predict a number of genes, depending on the threshold and the stringency. And we don't quite have negatives. I know maybe you're familiar with ROC curves. Well, we don't really have negatives, so we can't do a proper ROC curve. But this is just how many predictions we're making overall. And the y-axis is we had a curated set of about 70 targets for this pathway. And we wanted to see how well we predicted those. So being up here is good. Of course, those 70 benchmark set is not the only possible targets. They're just the ones that have been known so far. So you want to be somewhere in here. Each of the individual types are green, blue, and turquoise. So conservation, as I mentioned, is the worst. It's not the most informative to identify targets, although it's got some predictive value. Expression was better, but not as strong as we thought. Binding was actually the best in this particular example. Binding was very strong. And I think it's the way the CI is. It's a single transition factor in this pathway, and it's very direct, its role. The chain fit, so this is the model that was picked to be the best. That's the red line. So there's a boost in performance. There is a contribution of including expression and conservation. Okay? Binding alone is good, but you can improve more. And we looked at some of the top genes, and with the integrated model, we were finding pathways. So we looked at KEGG pathways in gene ontology that were very reflective of developmental processes that you would expect, and they were more enriched using the integrated model than just using each data type individually. So it showed the power of using an integrative approach. I had other examples I don't have time to show, but other transition factors in Drosophila yeast, another yeast example. And it's not always clear that this is the best model. Sometimes the layered model was picked. So in advance, you don't always know, at least based on the data, which maybe is a flow of information. So this is available as an R package called LC Mix. And it's a general model. It's for any number of data types. It's not limited to those three. Different distributions. We had one example that we tried that had count data, so it would be appropriate for sequencing data. Maybe long-tail distributions, so some of the non-traditional distributions, and sometimes that reflect the kind of array data or what you see. Multivariate, so we had one example, multiple binding data, multiple experiments, or multiple expression experiments. And we're working on a semi-supervised approach. This is considered unsupervised. We can't do supervised because we don't have a negative test set. But we can use maybe those positives, that high curated set of 70 examples to maybe help inform the algorithm, okay, to help prediction. And new applications, this has been applied, going back to this asthma example, where here they had methylation data and transcriptome data. Just quickly talk about this. There were about 100 thematics and 100 controls in blood. And a gene list approach studying each data type separately and trying to intersect had very little results, or almost none. Just had low power. So then they applied the integrative approach and were able to find many more genes, and they were related to some of the pathways they expect for asthma. So like I said, it can be used in other applications. So finally, I will now talk about, so this up to this point has been all about transcription, looking at gene expression, looking at binding, transition factor binding, and methylation, the role of methylation. The last topic is post-transcriptional processing. So I've grouped that together, although technically this is after the transitional process. So one form of post-transcriptional processing is controlled by microRNAs. Okay? So that's a current area of research for me that I've been excited about. So I'll talk about kind of how I got into this and some current projects and just sketching out the ideas. And here I'll come back to the alcohol research. I mentioned I have this long-standing collaboration with alcohol researchers in Denver and Boulder. So the motivating example here is these behavioral models. And the group I work with looks at alcohol consumption in these animal models, and they're interested in identifying the predisposing genetic factors for voluntary alcohol consumption. So one of the models that they look at. They do other ones like tolerance and sensitivity. And the data that we examine is there's genetic data for the different strains of mice. These are all mice studies, mouse studies, the transcriptome data, obviously the behavioral data, and all on, in this case, recombinant indrid panels. And my role is contributing to the genetical genomic techniques to identify candidate genes that predispose to this behavior. So I'll highlight one of the studies, the drinking in the dark model, that relies on the rodents' normal patterns of eating and drinking. So these animals are provided ethanol during their dark cycle, hence the name. And there was the behavior was collected on several strains and on a recombinant inbred pattern that we call LXS. So it's a cross between the long inbred and the short inbred strains. And I'll highlight one of the candidate genes that was sort of the motivating story. So this one candidate gene, GNB1, came up in several analyses of this drinking in the dark and also other models of alcohol preference and consumption. It's guanine nucleotide binding protein, beta-1 subunit, and it's important because it transduces signals from G protein coupled receptors. That's its role as a signal transducer. So here's one of the figures from the alcohol consumption work where they measured gene expression levels for pairs of strains. So the x-axis is pairs of strains. One is a high alcohol drinking, and one is low alcohol drinking. So red is low, that's the red. High is blue. And the expression, what's reported here is relative to the low alcohol drinking in red. So those are 100%. So you see a reduction in the high alcohol drinking mice in the levels of GNB1, transcript levels. For the most part, I'll get back to this anomaly here. When you look at protein levels, similar pattern. This is based on an antibody-based assay. Again, lower levels in the high alcohol drinking mice. So these are, for example, high alcohol preference selected lines. 1, 2, 3. These are classical strains. And this is parental strains of the panel that I'll be discussing. So as you notice, there's this one anomaly here. It's pretty strong. So it posed the interesting question. Why is this one different? And especially the protein levels bounce back, and they're consistent. What's reversing here? So I was interested in this because I was getting more into microRNA-related research because that could be one possible explanation, and there could be others. But perhaps there's a role of a microRNA here, or one or more. So do microRNAs play a role?
Does GNB1 regulate GABA transmission? Is that what you're getting at based on the title of the paper that you cited?
Yes. So it's yes. Here's this. It's related to the GABAergic system, and no, that's not. I'm getting more onto the role of the microRNA. So I know it transduces these signals from the G protein coupled receptors, and it has a role in this GABA system.
GABA receptors.
System. Yes. Yes. And unfortunately, this is not my expertise, so I focus more on the analysis. But and there are, if you want more details in the paper, they've gone into depth about the possible pathway and all the different mechanisms. So going back to microRNAs, this is a review. These are small endogenous non-coding RNAs. They regulate target transcripts, either promoting transcript degradation or repressing translation. So in this example, the hypothesis would be repression of the translation process, so that the protein levels would be reduced. They are transcribed from the genome, and they derive from transcripts that then fold back onto themselves. So you get this interesting hairpin structure here. Now, these, like I said, they target genes. And as a review, they target genes typically in the 3' UTR. So here's an example of the targeting. So there's the cleave version, which is short, 10 to 20 bases. And there's some base pairing, although it's not perfect. And there's some secondary structure involved with the actual target binding. Now, there are experimental resources collected based on, there's different kinds of assays to look at binding. GNB1, so that's the candidate gene, is not in any of these experimental databases. So there is not an known, at least, role of any microRNA for regulating GNB1. And these are collected from human and mouse resources. they're computational-based methods. So they look at the biophysics of this interaction. They look at the base pairing, again, allowing for some mismatches. They look at conservation across species. And they come with a metric for prediction. And there's different algorithms. I won't get into them. There's at least a dozen that are out there with nice databases that they provide computational resources. But if you go to any of these, I don't know if any of you have, there are thousands to hundreds of thousands of predictions. There's a lot of information there, a lot of predictions. And there have been compilations to try to make sense of those, to put them together. They tend to rely on set cutoffs, but they also tend to be web servers, so they're not very necessarily convenient to do a lot of the downstream analysis that we would be interested in. So this sparked a side project. A lot of the microRNA research was relatively more in cancer. You don't see as much in alcohol research. There's one very nice study of miR9 in the alcohol research community. It regulates, it was found in rat striatal neurons and regulates different splice forms of the BK channel that have different sensitivity to ethanol. And it's a very nice story that's been followed up with multiple molecular studies. And that's probably the best well-known study in the alcohol research community. And like I mentioned, a lot of it's in cancer. So there's more in cancer. So a lot of these databases are also biased towards more cancer-related resources. Now I had, because of the cancer connection, I started to interact with some of the cancer researchers who were studying microRNAs. And they were coming across the same problem I was, and about the usability of some of these resources. So I had my own compilation, and I worked with a postdoc in the Cancer Center to take that and create a R package that can be usable and to facilitate this type of analysis. So this was sort of a side project. It's under revision right now, and it collects millions of records, different databases. It has drug and disease information in addition to the computational and experimental resources. It also allows for more control in that we try to have user-defined cutoffs so that we can control maybe the strength of those predictions. Every algorithm has its own threshold and type of summary. So we wanted to try to look at and try to control that ourselves. So previous version of that package, I'll return now to the GNB1 story. Okay, let's go back to the GNB1 story. So here's a snapshot from UC Santa Cruz showing the location of GNB1, the UTR. So this is the UTR in the genome. And what we're interested in is where is this probe set? So here's the probe set. So it was an Affymetrix array that measured the expression data. So the probe set actually was occurring that showed differential expression in the UTR. Okay, so it's important to go into more detail to find, especially with these arrays, where are these probe sets located. So it's actually in the long form of this UTR. There's a shorter form as well. And based on this compilation I had created, there were multiple predicted microRNA sites supported by multiple algorithms. And these were the 218 and 101AB. The hypothesis was that perhaps one of these two microRNAs are targeting GNB1 in the high alcohol drinking strain to repress translation so that protein expression then was reduced, and then you had consistency with the other types of strains. So I worked with the microRNA guru on our campus. She's now since left. Again, she worked in cancer, but she had a special QR2 PCR assay that she had developed to look at microRNA expressions. That was the first question: is, well, are these microRNAs expressed in these brains? This is all brain, by the way, all brain of these strains. And Richard Radcliffe and Beth Bennett were very generous in giving some samples, so I could coordinate with Lynn, and she looked at the microRNA expression levels. In their model, they had the strains were ethanol and saline treated, but we also had naive, and we could look at the strain effect. So we really wanted to know the strain effect, not the ethanol treatment effect. So here's some of the preliminary results looking at strain effects for these two microRNAs. We did see a relative increase in the expression level of the microRNA in the high alcohol drinking strains, okay, which was the hypothesis that we proposed. They barely survive the 0.05 threshold, but we thought this was good for pilot data, and there was almost a twofold change. So this helped support the... Oh, yeah. what we would have more power, I guess, to see more. Well, yes, so we don't have much power. This was based, and it was also not ideal because we had other ethanol and saline treated, but unfortunately that was the I could get a hold of. So if I would do this again, having a larger N for dust, the naive in the two strains. So I would design. You would have more. You would have more power. Yeah, we had more power to detect these smaller differences. Okay. So it was more of a pilot study. There were some limitations because it was just what we could get at the time, what was given to me. So I think it would help motivate a larger study. And that's just a summary. You have the high alcohol drinking mice, longer form of the GNB1 transcript, and then at least suggestive higher levels of the microRNA. What we're working now with Lynn is, I've done this in the COPD study, is looking at a binding assay. So that would be the next test is this microRNA. So it seems to be expressed, and maybe we can improve the data and improve the study. But let's also look at binding of the microRNA in the UTR, the GNB1. So there's a luciferase-based binding assay that I've done with the COPD study in some other candidate genes for lung disease. But the idea would then to do that next year to now look at the binding, the actual binding. So all this sparked my interest to, I like that story. It was one story, one candidate gene was, well, maybe we could find other examples like that. So the idea was to look at the entire panel. So this is the history of how this panel was determined using this bidirectional selection for differential sensitivity to sedative effects of ethanol. So there's a whole panel of up to 60 strains derived from those parental strains, the ILS and ISS. And there's extensive genotyping based on a SNP array diversity panel in mouse. There's now, Richard Radcliffe has a whole genome sequencing of the two parental strains. There's dozens of alcohol-related phenotypes. I'm focusing here on alcohol preference, but there's also sensitivity and tolerance. And there's the whole brain gene expression available. And again, Richard's doing RNA-seq on the panel. He has another model which includes ethanol, saline, and ethanol-treated mice. So the goal that we received funding for was to now profile microRNA, do this more systematically, particularly this case study, and scale up, profile multiple microRNA to identify other potential microRNA-regulated pathways like that. So we had microRNA and the gene, which would be associated with the predisposition to alcohol intake, like the phenotype I talked about, but also response. And I've just completed some microRNA expression profiling. So this is small RNA profiling using Illumina HiSeq on the six of the strains, just choosing some of the extremes to start with, and hopefully presenting that at RSA for those of you who go there. But the computational methods are how do we integrate the microRNA expression. We have the gene expression, genetic data, and all the behavioral data together. And this is what this slide shows is, here's one example phenotype, the distribution of the loss of writing reflex, where you have the parental strains, but then there's a spectrum from all the other strains. So you can see a pretty wide variance here in diversity of this particular phenotype. The data can be represented here. We have gene data, microRNA data. We have these interactions between genes and microRNA based on that multi-mirror package that compiles that. You can look at protein-protein interaction. There's all the genetic data. And some of the methods have been developed to look at gene and microRNA expression and look at negative correlations. Some of those methods are not so focused on the disease or the outcome or the phenotype. So I think emphasizing that would be contribution and linking it with all the genetics. So that's a sketch of future work. And that concludes my talk. I'd like to wrap things up. I'm a little over. So these are group members, students, and research assistants in my group. I didn't talk at all about the COPD data, but I have some students working on that. Alcohol researchers, and I also forgot to put Beth Bennett. She's been she's taught me a lot about some of these behavioral phenotypes, and various collaborators I've mentioned throughout the talk. And finally, my funding from a previous K grant and now an R01, and support by an NLM training grant for many of these students. So thank you for your time.
So we're about at the hour. We probably have time for one or two
Sorry,
questions.
no, I remember.
Please press the microphone button down so it gets recorded.
I'll do a quick one. In the SULDEX method, did you ever run into a problem of degenerate sequences after the transcription factor is bound to some sequence, you pulled it down? Did you ever have trouble mapping that to the genome, like it mapped to more than one place?
That was not, those were never bound, never mapped to the genome. It's in vitro, so it's just looking at the theoretical properties of binding. So it's, again, in theory, what would this transition factor bind to? Now,
They're random sequences.
yes, they're random pools of oligos. But that's important because in the genomic context, that could affect binding. And some transition factors have cofactors, and so you could affect local context in the genome. So that's why the in vivo binding data is important, although that resolution is not, it's several hundred bases. So you have better resolution with in vitro assays, but more genomic context, but less resolution in vivo. So they both have their pluses and minuses. Okay, thanks.